Thank you and welcome to this last session where we have devoted 4 talks presentation to Baltic and Nordic barley from a regional perspective. You should keep in mind there are not so many people living in this area, but we have a climate which make it less interesting for big breeding companies. Still, we have 3 breeding companies in Denmark with a big program on barley. There's one in Sweden. There's also in Finland, and we learned the other day, there's one single barley breeder in Norway. And then also
Iceland.
here. Sorry?
Iceland, you mean?
Was that in Iceland?
Norway.
There's one breeder, yeah, for 2 years at Graminor. There's one barley breeder in Norway. Basta. I think we'll hear more about that, and we'll have some examples from the first keynote speaker, Teresa Bengtsson, who is from the Department of Plant Breeding at SLU in Sweden, Alnarp, and she'll talk about Together We Are Stronger, United Forces to Promote Nordic Pre-breeding in Spring Barley. Please.
Thank you. And thank you for the organizing committee for inviting me to present a decade of work. So, yeah, and I think the title also, I just have to say, it's quite suitable now when yesterday they signed the application to NATO for Sweden and Finland. So together we are stronger. So, as Søren mentioned a little bit, we have quite a unique climate in the Nordic countries, or in the region. And first of all, we have a north to south gradient. And with that, I'm referring to that we have an increasing growth period. So it's actually quite a big difference between the northern, if you take for Sweden, northern Sweden and southern Sweden. It can be almost 100 days difference in the growth season. So, and we also have a gradual change in temperature. And this is not enough. We also have a west to east gradient, where we have the maritime to continental climate. So Iceland, for instance, we have very cool summers and mild winters, whereas we in southern parts of Sweden and Denmark, for instance, we have hot summers and cool winters sometimes, but more in the north of Sweden and Norway and Finland. And then, more importantly, we also have very long days. And now in June and July, the sun hardly sets at all, especially north. So that also makes it difficult to grow more southern European cultivars in our countries. So why do we then need a Nordic public-private partnership? First of all, just as in other parts of the world, we are facing the climate changes. We have increased temperatures. We have seen more occasions of like spring drought, warmer autumns. And there is also new pests emerging. And we have seen that very recently, that we have problems with a new pest in the wheat, for instance, a gout fly. And still the day length remains. So we have to combine the breeding for both the future climate and also for the long days. And as you all know, food security couldn't be more reality or actual that it is nowadays when we have the war going on. And the market in Nordic countries is very small, and there is, since the 70s to the beginning of this 2000, and over these 30 years, there have been a very decline in the number of companies. So going from like 16 to 10 companies, breeding companies in agronomic, in agronomy. And then also, we have also reduced the number of programs. So not only the companies, but also the programs and the funding for this. And then we are dependent on cultivars breeded from the big companies that is not aiming for this small market when they breed. And then we needed to then increase the competitiveness for our company, our small companies, to be able to compete with the larger companies. And no one else will do the job, I would say. So, and then the next question. So now we know why we should go for public-private partnership in the Nordic countries, but why in pre-breeding? The most obvious answer on that is that it's pre-competitive. To have a lot of competitors collaborating, it needs to be pre-competitive. And it's also maybe the area where you don't have the same resources in the small companies to do. So with the pre-breeding here, we are referring to germplasm evaluation, but also to introducing new traits or genetic variation in adaptive background. And also very important to develop new tools and test out more high throughput phenotyping equipments and more expensive or new emerging tools. So based on this and a report that came out, a report that came out with the Nordic Council of Ministers in 2010 that was written by Roland von Bottmer and Anders Nilsson, where they pushed for this, that we should have an investment for a public-private partnership. Based on that report, 2012, there was an initiative from the Nordic Council of Ministers to have a platform for this public-private partnership. And so in 2012, I started a few projects. One of these was a barley, which I will present here, but there was also one on apple, one on ryegrass, and one on plant phenotyping. So the money for this project is coming 50% from the Nordic Council of Ministers, and the other 50% should come as, from in-kind from the private industry. And then the administration is handled by Nordic. And for the Barley project, we have Ahmed Yahour here, that was a project leader. And he's head of breeding at Nordic Seed, but he's also adjunct professor at SLU. And it was quite beneficial to have someone that comes from the academic and then is working in the breeding companies, because he could understand the, both sides of the coin, so to say. For the Barley project, we have the private partners. So we have where they have breeding, so now we can make that clear. So it's Graminor in Norway, and we had two companies in Denmark, Sayed Plant Breeding and Nordic Seed. And then we have Boreal in Finland and Lantmännen in Sweden. And then we also have from the public, also I'm putting in the middle here of the public and private circles, we have the Agricultural University in Iceland, which also have a breeding program for barley. And then for the public partners, we had Luki in Finland, SLU, and Copenhagen University. And Copenhagen University in Lantmännen was in the first 3 years of the project. So a brief outline how this project was, the activities of the project. And as I said, it started 2012 and it lasted until 2020. So it went over 9 years and it's divided into 3 phases. And the first phases were screened adapted material. And the aim was to get linked markers that the breeders could use for market-assisted selection. And then for the second phase, we instead added more germplasm and then more historic material and land races. So about 200 lines were screened for different traits. I will go more into the details sooner. And then the material, we were also producing medic populations. So the aim here was to have material for the breeds that they could continue to use in the breeding programs. And then the last phase, we wanted to screen all of this material we have developed in the second phase, and to deliver linked markers for these populations that could be used for market-assisted backcrossing. So when you're working with, I think many of you know it, when it's academic and the private partners, there can be problems and you need to understand each other's agendas and what you need to what is counted, so in publications in the academia and other things in the breeding. But we also now have had several competitors in the project. So the first year, it was really a lot about building trust and mutual understanding between the partners. And then when that was done, we could start with the real action. So, first phase. So here we had screening of adapted material. So each breeder provided 30 lines for breeding companies. So it should be breeding lines of modern cultivars. And these were screened in multi-locations in the Nordic countries, but also in Germany. And they were screened for several different traits, so several different diseases, and also for development traits, height, straw strength, and some yield components. And all of them were then genotyped by the 9K, which was available at that moment. And then we were conducting GWAS with the aim to find markers that they then could develop CASP markers of in the breeding companies. And so one part of the project was also to develop these CASP protocols and validate the markers, and then these protocols were shared among the partners. And at this time, not all companies, as I remember it, had the CASP protocols set up or used CASP yet, so this was a great advantage for them to start up and have a set of markers to continue with. So one example, we were screening for the cereal cyst nematode, and this was outsourced at this time to a Danish company, and it was a lab-based method for the screening. It was very expensive, so they had 2 years of sending the lines to this company and screen them, and this resulted in that 2 marks were found on chromosome 2H. And you see that in the table here is a marker 1 and marker 2 to the left, and then you have number of lines and the average infection. And the one in green highlighted is the only one that is really resistant here. We have a scale 1 to 9. And so the results showed that we had 2 markers, and you needed both of them to have fully resistant lines. And these markers are now used by the breeding companies and has been very successful. And they are now able to screen at an earlier stage and also more lines than before. So just an overview, I don't expect you to see all. Each color is a different trait, but a lot of markers or QTLs were identified, and CASP markers was developed by the companies and validated and are now in use. And then, of course, for the academia partners, it was also important with the publications. So we had a few publications on looking into the genetic diversity and structure and LD of this germplasm, and one looking into the GWAS results from the mildew. And then we have Magnus Göransson here that will talk later today. He was looking into the trait earliness because it's very important for Iceland, where you have a very short growing season. And so there was one paper from that, and there is still plenty of data and some manuscripts in progress. So after phase 1, then we got the knowledge about the material and markers and the link to different traits. And then this marker could now be used by the breeders. So then we moved on to the second phase, where we collected 200 genotypes adapted in historic material and material from Nordic, among others. And these were screened also at different Nordic locations for 2 years for disease resistance traits and agronomic traits. And while we were doing this, we in parallel started to develop medic populations. And these were based on the results from the first phase, where we screened the adapted material, but also on the second phase, where we screened the more historic material and land races. So these medic populations, they were balanced for row type, since we have breeders both interested in the 6-row and breeders interested in 2-row barley. So they were also balanced for advanced materials, so 50% should be more advanced material and 50% could be more historic material. And we had a couple of medic populations with 8 parents and some with 4 parents. And then we created double haploid lines, and everything was multiplied in New Zealand. So here is a list of the medic populations. We had in total 9. So the first medic 1 to 5 is with 8 founders, and the 6 to 9 is with 4 founders. And then we also created 2 biparental populations. And we pyramided them for different traits, different resistance traits. So the first one is for scaly rust, net blotch spot type, and so on. And then medic 5 was the only one not for disease resistance. It was for earliness, and it was developed in Iceland. And we aim to get about 200 DH lines of each population, which we were not successful in. As we have mentioned before, it's very genotype dependent. So in the later populations from medic 8 and the biparental, we instead used single seed descent. So from the second phase, we retrieved this material, a lot of populations, and that was pyramided for resistance and or early maturation. And this material was all divided among the breeders, so they all have access to it. And then we went on with the third phase, because the material populations is not so much of value if you don't know anything about them. So we continued to screen these medic populations, and they were all unit with a 50K Illumina chip. And then here is very small, so, but each of them were screened for the diseases they were pyramided for, but also every disease that was occurring in the field that season, to collect as much data as possible for them. And also days to heading and days to maturity and plant height was collected. And the last populations were completed quite late, so they are not screened as much as the first 4-1. So I will take an example of one of the publications with Hautsalu et al. For the SCALD results. So here we have used medic population 1 to 4, and they were screened at, in Finland and Iceland between 2017 and 2019. And the distribution you can see here, so it's medic 1 and 2 and the blue one, and then 3 is yellow, 4 is green, and then the pink one is all of them combined. So medic 1 and 2, if you remember from the table before, they had very few lines. And the difference, if you looked in the parents between them, was only 1 parent. So we decided to combine them for the analysis to have enough data points. The structure, if we looked in each of the populations, there was none, which is expected for the medic populations. On the PCA to the left, you see a PCA over all medic populations together, and there were not much structure there either, but you could see them clustered according to the populations. You will have difficulty to see it, but I will see if I can try to show here. I can hardly read it here. So in the medic 1 to 2, the red boxes highlights the one that was resistant. So all the lines below, or the genotypes below, is the parents used for the different populations. So for medic 1 and 2, we, when it comes to scholar resistance, we had 1 Jordanian landraces that was resistant, and then we have our favorite here that we talked about a lot, Planet, and then a breeding line from Sayet, and Ylittonian and old Finnish landrace, I think. And also for medic 3, it was the only parent that was donor here was a Planet. And then in medic 4, we have a Danish landrace, Gaffelbig. So here you see the QTL allele combinations of the most significant QTLs. And if there was any QTL or allele combination that was not present in more than 5 lines, they are not included in the graph. So the color is the same here, so it's medic 1 to 2, and then goes on. So you could see here that we seem to be able to pyramid different alleles in the same lines. And the most effective QTL was the one on chromosome 6. And it was found in Gafferbygg and also in the Jordanian landraces and planet. And then, let's see. Just a brief overview of the QTLs detected. So you have the QTL in green and other QTLs reported in literature in blue, and then novel QTLs in red. And the one here in red was actually a report of that now in 2022 from Claire et al., I think it was, and that have screened Turkish isolate, Rhynchosporium isolates, and 5 out of 6 of them had this QTL showed resistance towards 5 out of 6 of these isolates. So it seems to be working also against Turkish isolates. So we're still in phase 3 now, the last phase of it. And except for the screening of the medic populations that we developed, we had a new work package where we wanted to look into early vigor in adapted and historic material. So we got an EPPN 2020 grant in collaboration with Dr. Kerstin Neumann at IPK. And there we included the 180 adapted material from the first phase. And then it was not balanced for row types, so we included more German historic cultivars, but also land races and cultivars from the Nordic, and some more breeding material from Finland and Denmark for 6 row. And then we were screening it for 3 independent experiments in their Levnatech facility, where we looked into the digital biomass over time. And yeah, you get, as if you ever use them, you get a lot of other traits as well, up to 30 traits, I think. But what we were interested in most was the digital biomass over time. And we were also looking into photosynthesis and chlorophyll content. And some preliminary results. This is manuscript that's we are writing on at the moment. Here we have on the graph, you see a Manhattan plot over a multi-trait GWAS we have done. So in the green dots here is traits that we looked into in the Levnatech facility, so the digital biomass and NDVI and some other parameters from the photosynthesis. And then in blue, you can see results from the field, because we had to multiply the material first to have ensure that we had the same age and background of the material before the experiment in Germany. So then we took the chance also to look into the 1,000 kernel weight and number of infertile spikelets, and then, or infertile spikes. And so the blue is the result from that, and then the pink one is when we combine all the traits, both from the Levnatech and the field. So we see here that we have something, 2 QTLs on 5H, that is really increasing the more traits we included here. So the results from screening of the medic populations 1 to 4, which is screened mostly, is here shown for the resistance traits, for the biotic stress. And then there is also, at the moment, there is 2 publications with this, the SCALD, and then also a mildew manuscript. And we saw also that there was some lines that had several alleles, favorable alleles for both mildew resistance and scald resistance. So these lines are very interesting to move forward with. And then we also have one important part with this public-private partnership is education of next generation of plant breeders. So we have also had master thesis projects, and one of them was to look into the GWAS of these medic populations for leaf rust. But we also had for the material screened at IPK, we were also having a hydroponic trial where we looked into the root length of the seminal root length. So here is the results from the earliness population that was screened. That's also a manuscript ongoing that I hope will come soon. And then and that's here in blue, the QTLs, and then we have the green, it's the results from the IPK study. So from phase 3, we are retrieving this markers that is now linked to traits in the material that the breeders have got. So now they can use the material plus the linked markers to move on if they want to include them in their breeding programs. But this is not all, of course. Other benefits with this kind of project is the strengthening of the Nordic network. And it's been it's been very successful, and all the breeders and the researchers have been very happy about it, and we have now continued the networking and tried for getting new grants. So I hope this collaboration can continue. It's increased exchange of knowledge, but also material, and access to new genetic resources for the breeders. And the possibility to use this large-scale field and in different environments is a great benefit. And use of new techniques and methods that was not available before, like the CASP system that is now up and running everywhere. And then possibility to use this more advanced crossing scheme with land races and more exotic material. And then, not least, education. Because if it's something we noticed, it's that, and the breeders is talking about, is that it's very difficult to get find new breeders or people that wants to come in and work. So that is one of the benefit. And then all of the material and all of the information is saved in our database. So we have internal website where we have shared all the informations, literature, protocols, and so on. And this is still up running, some time at least. And yes, so I think I will round up here because I see the time is. But I would like to thank the two men on the lower right corner. It's Roland von Bottmer to the left and Anders Nilsson to the right, which was the man behind that we got this public-private partnership in the first time. So thanks to them and all the project partners over the years from 2012 to now. Okay, thank you.
Thank you for the opportunity to present historical overview of spring barley breeding in Lithuania. And the cultivation of spring barley has long-standing traditions in Lithuania because spring barley is one of the predominant spring cereal crop in the Lithuania. Barley is superior to other cereals by the diversity of biological types, ability to grow in different conditions, duration of growing season. But as is the whole Europe, the area of barley crops in Lithuania tends to decrease, but remain one of the most important. In this slide, you can see area of spring cereals in the last 20 years. And in 2000 years, spring barley area was 350,000 hectares, and now for 2020, it is 140 hectares. And in the last 6-7 years, it is overtaken by spring wheat. But in 90s, spring barley area was 500 and 600. The yield of spring barley is a second among spring cereals, but in last 4 years, it is similar like winter wheat. And the beginning of breeding starts in 1922, and spring barley varieties have been developed in Lithuania for 100 years. The idea to establish a plant breeding station in Dotnuva, according to Professor Dionysas Rudzinskas, proposed agronomist Juozas Valiūnas, who at initiative of the Ministry of Agriculture visited Moscow breeding station and met its founder and manager Rudzinskas. Situation, political and economical, was suitable to return to Lithuania, So on 1st of March, an order was signed on the establishment on the Dotnova breeding station at the already operating agricultural technical school, appointing Professor Rudzinskas as the head of the station. The first head of Dotnova breeding station had a lot of professional knowledge and practice experience, was acquainted with the experience of plant breeding in the Western Europe and the United States. And arriving in Dotnova, he brings with him the genetic collections he has collected since 1903 and the lines and varieties created as a Moscow breeding station, which later became the basis of the first Lithuanian varieties. And barley breeding starts in 1922 and non-stop. In meantime, other cereals breeding was stopped and restarted. Now, the cereal breeding department of the Lands Institute of Agriculture carries out breeding program of spring barley. And the main task and purpose of breeders are development of spring barley lines and populations, investigation of their adaptive properties, elements of yield structure and agronomic value, development of new promising varieties, and maintenance breeding and seed production of those varieties, and the selection of the most suitable varieties and breeding lines of spring barley to organic farming. The basic milestones for barley breeding can divide in 4 periods. First initial period, elementary breeding in 1922-32, period of improvement of breeding 1932-1972, and period for progress and intensity of breeding 1972-1990, and period of breeding in global context from 1990 to present. At the beginning of the Dotnova breeding station, efforts were made to preserve the barley collection collected by Rudzinskas and to collect more initial material. And in 1922, the barley catalog entered 43 local samples and 98 samples from Moscow breeding station brought by Rudzinskas. In 1924, agronomist Jonas Bulavas, later professor of Lithuanian Agricultural Academy, joined the rank of breeders to start breeding work in true sense of the word. And at the beginning, breeding work main method was individual selection. It was desired to distinguish pure lines from local or other varieties. Since 1928, in addition to the individual selection method, a hybridization method has been introduced for the development of new barley varieties. During this period, 5 varieties of spring barley were developed. Auxiniai was the first variety and made from developed by individual selection from Sweden variety Gull. Number 2, plikieji, or the naked, and gulbės kaklas and swan neck was made from selection from local land races, but were not widespread. Dotnuvos ketureiliai was only one 6-row Lithuanian variety, and džiugiai was tolerant to acid soils and was registered for 25 years. In the second period, researchers of Dotnova breeding station actively interacted with the breeders of neighboring countries, exchanging plant genetic resources, and in cities, the work of breeding station was significantly advanced by the internships of the station staff in the breeding institutions of Europe. One of the most important business trips abroad was Zigmas Mackevičius, the newly designed Dotnova breeding station director, long-term secondment to Sweden and Denmark, running the entire vegetation period, and after that, breeding methodology was improved based on the experience. And a special seed reel for breeding was introduced. You saw it in previous slide. And Barley breeding since 1932 moved to more objective evaluation of breeding material based on trails data, and much consideration also given to material obtained from other regions in the Soviet time, from Soviet Union and from abroad. Several hybridization variants were used to develop new barley initial material: cyclic crossing, backcross, intravarietal crossing, and free intervarietal pollination. The method of mutagenesis was also tested for the development of the new barley varieties in 1959, 1964. However, hybridization with subsequent individual selection remains the main barley breeding method. Barley breeding was carried out by Bulovas for 34 years, and after that involved new breeders like Subačius in the World War second time, and post-war time Paidokaitė, Leistrumas, and Terlauskienė. In the second period, there was developed 4 spring barley varieties, and 2 of which were registered, and very successful was variety Auksiniai 2, and it was grown and registered for 40 years. And how you see in the pedigree is varieties Kenya and Isaria. About these varieties yesterday talk in the third period. Since 1975, further breeding center was established in Dontuva with new laboratories of genetic, plant physiology, and plant immunity, and with greenhouse. It increased laboratory input affected barley breeding too. F1 cultivation and hybridization are started in the greenhouse. More modern breeding equipment for sowing and harvesting was purchased, and decreased level of mechanization affected the increase of breeding extent. During this period, there was developed 6 barley new varieties, and 5 of them were entered national plant variety list. After Lithuania stand independent, there is a need to develop homozygous varieties because the US tests were introduced to register varieties. After internships and courses abroad, the pedigree breeding methodology has been improved, and we moved from the selection of individual plants in early generations to selection of spikes. In 2009, modernized greenhouse helps to shorten breeding process. It carries out hybridization and grows the F1 generation. You can see spring barley breeding scheme. It is initial material nurseries and mass selection hybrid nurseries F1, F2, individual selection hybrid nurseries F3, F5, and from F6 we starting to evaluate yield and other economical valuable properties. In this period, increased contribution of biotechnology, and the first Lithuanian dihaploid spring barley variety, Alisa DS, was developed. Unfortunately, this program was later closed. Growing international cooperation, expanded breeding material testing with breeding companies and colleagues from Latvia and Estonia. We participate in international projects like POST, SUSVAR, Hordeva, and now we involved in the Eva Wheat and Barley Network and Baltic Barley Consortium. We also cooperate with colleagues from institutions of our country, and we have been testing barley mutants created in Vilnius University and have some publications. An important source of the initial material is collection of genetic resources. A large collection of spring barley genetic resources has been accumulated in the cereal breeding department from 1994. It consists from varieties of Lithuanian origin, varieties from active walking collection, and breeding material from different breeding stages. Now, number of samples in our genetic resources collection is 1,200 48 samples, and 115 samples are placed in Lithuanian plant gene bank, and 20 of them are duplicated in Svalbard and seed vault. The selection of the most suitable varieties and breeding lists for spring barley for organic farming is being carried out. Promising lists of barley have been studied in conventional and organic backgrounds. Phenotyping are based on the response of genotypes to agrotechnical level, disease resistance, and natural weed control. During this period, were developed 12 spring barley varieties, and 7 of them are entered Lithuania plant national list. And during whole period barley breeding, 27 varieties of spring barley were developed in Dotnuva, and they have been registered and cultivated in the country for various periods. Lithuanian varieties cover about 15% in the national list of plant varieties. And contribution to the national budget, proportion of Lithuanian varieties in total spring barley area covers only 6%, but in organic area covers 53%. And value of total yield with LAMS varieties is about 6 million euros. The most widely grown Lithuanian spring varieties are Emadeus and Noedeus, and the most common Lithuanian spring variety on organic farm is Alisadeus. Main goals for spring barley breeding program in the cereal breeding department remains productivity, resistance to diseases, resistance to lodging, and improvement of grain quality for human consumption and feed. And for the future, immediate problems of barley varieties to be addressed by breeders are to create particularly high-yielding varieties in the traditional format with quality gradation that maintains the criteria of competitiveness, varieties suitable for Green Deal farming and ideas with increased pathological resistance, more efficient use of water and nutrients, reduced need for chemical preparations. Also, varieties meeting the requirements of the new EU organic production regulations, meeting UPOV standard requirements for special organic varieties, multi-line varieties of related genotypes, and varieties suitable to narrower users but more specific as functional foods, specific uses according to industry and market needs, and to prepare and use a genetic pool of varieties and diversity for the development of basic research. So plant breeding is a journey, not a station of arrival. Changing territorial conditions and requirements for plant biomass are demanding ever new plant genotypes. Thank you for attention.
The national and local breeding program of any crop is of great value for any country, and especially for a country like Latvia. This includes genetic resources and diversity, the creation of varieties capable to adapt to local conditions, accordingly producing added value for merchants, and their potential to be used as a base of applied and fundamental research, and also for growing up a new generation of scientists. Today I'm going to guide you through the information about spring barley breeding program in Latvia and also research. In hope you can find the information beneficial, seeing the potential of our region for future collaboration and maybe also for betterment of your business. We have many of Latvian songs that tell about growing barley and processing of brewing beer. Barley was the first crop. It was mentioned that what the first crop that farmers grow in Latvia. Accordingly to researched information, Four-row, two-row barley was the most frequently sown in Latvia in the second half of the 18th century. Six-row hooded barley and armless barley were also mentioned. From the beginning of the 18th century, seeds were imported from Germany to improve barley yield. However, details about the varieties used can only be found later. There was mentioned Chevrolet and Phoenix barley were introduced on the minor farms. The so-called Himalayan barley has also been grown. Farmers called four-row barley and six-row barley like as local, while the two-row barley was named and considered as a German barley. The first barley variety trials were set up simultaneously with the foundation of research center located in Stend and Priekuli, and 2 years later also breeding programs were established. Over the years, spring barley breeding was done based on the specialization and in close cooperation by realized the different barley research projects in these 2 institutions. About 80% of the total harvest is used for animal feed in Latvia. Starting 2014, the farming cooperation Latrap started operating a malt factory in Latvia. It has the potential capacity to cover about 25% of the overall demand of malt in Latvia. This has also led to a greater demand of barley varieties with the appropriate malting quality, as well as it has provided also the new opportunity and challenges for Latvian farmers. According to data, almost 25% of the total beer production is generated by small breweries. Although as I have learned that in Latvia, only 3 breweries produce beer from locally grown barley grain. And Latvia is mentioned as one of the European countries which historically has long tradition of using barley grain in its diet. The market for barley processing products produced by the biggest manufacturers extends from far from our country. Therefore, also the production of high quality grain raw material is essential and therefore is part of our research. Keeping in mind the priorities such as yield and stability, the resistance to lodging and diseases, grain quality criteria for malt, feed and food purposes, and specific criteria for organic farming, in total 25 barley varieties have been developed. Currently, our list of plant varieties includes 11 spring barley varieties developed in Latvia. In general, Latvian varieties are medium input with the appropriate plant length, straw strength, and ripening time suitable for growing also with clover, as undersown crop and in the mixtures with peas. 2 varieties are Halles. Type covered varieties are suitable for feed and use. A range of varieties developed till the beginning of 2000 at Stend was recommended directly for malt production and used by local breweries. Today, if the protein content and extract yield are adequate, then for instance the variability index for these varieties unfortunately doesn't follow the standards set up our malting factory. Here I mentioned I defined a variety Abava as an unique variety because registered in 1980, its variety is still growing in our fields, and we are preparing still this breeder seed. Spring Barley program in Latvia is realized in a frame of the project that is supported by Ministry of Agriculture. In last 5 years, it has been delegated also by farmers' organizations as well, and Latvian Organic Association. Agreement about this project realization is signed each year. Here I would like to mention also our long-term collaboration in the frame of this project with researchers from Latvian University Laboratory of Environment Genetics who create for our breeding program double couplet lines. The latest released covered barley varieties from our breeding program are Feed Barley, Middle Early, Humor and Dizis, which are characterized with good potential and yield potential and stability, as well variety Saule. In Latvian, this means sun. Is early ripening with catenate protein content showing high resistance to lodging and powder mildew. The latest released thus far several projects were realized in close collaboration among breeders and geneticists from University of Latvia. One of them was the project where Latvian barley diversity, evolution and breeding optimization by modern biotechnology methods were carried out. In the frame of this project, the structure of MLA resistance loci in Latvian barley varieties and MLO resistant loci in European varieties and breeding likes were characterized. Methodology for assessment of pre-harvest sprouting were adapted, and evaluation of results of barley diversity on the trait obtained. And there were also trials where agronomic traits significant for adaptation to climate change in Latvian breeding programs were evaluated. Based on the data, almost 200 barley genotypes, including covert and hairless ones in 3 years and 2 sites. Thanks to focused research done by barley breeder Linda Legsdiņa in Priekuļi, organic barley breeding was established in Latvia as well. In this project, genetic diversity characterization of Latvian Spring Barley Association mapping population was carried out. And for this developed Spring Barley real mapping population, phenotypic results for beta-glucans, pre-harvest sprouting, fusarium, and blight were obtained. And the variability of Spring Barley traits essential for organic farming in association mapping population were also investigated. And by whole genome association mapping analysis, naked grain locus node was identified as a determinant of beta-glucan content in barley. One of realized projects was dedicated to the genetically diverse varieties, including genotype, mixtures, and parent and populations. And it was concluded that variety mixtures could be used as one of the appropriate approaches to stabilize yield. Allelic composition assessed by SSR markers showed that the cultivation of subpopulation derived from the 2 crosses over 6 generations concurrently under conventional and organic conditions did not significantly change genetic diversity. Organic heterogeneous populations for all crop species are currently becoming more accessible to the farmers also in Latvia through legislation coming in force since this year. Mirga, the first composite cross population that was created in this previously mentioned project was derived from DLL crosses between 10 parents and characterized with good yield potential and its stability. The last completed project aims at the evaluation of the agronomic performance of CCPs compared with mixtures and their parents and homogeneous varieties. Several methods targeted to reduce the workload of CCP's development and also to improve existing populations were tested. And the results you can still see in the poster number 42. The focused Halles barley breeding work has produced the first practical results during the last decade in Latvia. Currently, 2 Halles varieties, Irba and Cornelia, are offered for farmers and food manufacturers. At the moment, in Latvia, we cannot yet provide a continuous and large amount of grain material of Halles barley, which is required by large processing companies. And therefore, Halles barley is still considered in our country as a niche product, which is highly valuable by small entrepreneurs. Commercialization strategy has worked out and close collaboration with farmers, grain processors and food producers was established in the project with the aim to commercialize of research results. This project includes in-depth research to clarify the growing technology for this very early high protein and beta glucan variety Cornelia with characterized with very average yield potential in some years also sensitive to lodging with the task to precise grain processing technologies to be used as a whole grain material for food, like food grain material. Several new food prototypes was also created and I hope you enjoyed also with Cornelia grains in these days added to our meal. And the comparative evaluation of different Halles barley varieties in our conditions you can see in the poster 39. To speed up breeding process, more precisely the evaluation of promising breeding material is fruitful thanks to our collaboration among barley breeders. Reciprocal exchange of spring barley lines and varieties for evaluation, or so-called in ecological, it means without fungicide application, multi-location trials are established already 20 years in all Baltic countries. And during last year, the Baltic barley breeders from Sweden found such collaboration useful as well. Opportunities to speed up a breeding process now is available thanks to new greenhouse facilities at Stende, where hybridization work and multiplication of the first generation hybrid material now is carried out. Stende location, it's close being to the Baltic Sea and the Gulf of Riga creates good conditions of the development of various barley foliar diseases, powdery mildew, net blotch, also scald, and in last year's leaf rust usually show rather high infection level. We are providing data of the field observations for the European PGR evaluation network as well. And this year we established a comprehensive trial of Baltic spring barley evaluation network at Stende. And I am regarding these activities as an opportunity for the exchange of knowledge and providing the collaboration in the future. Breeding activities rely heavily on the classic approach, which is a time and resources consuming process. In order to fully employ the benefits of genomic selection, a fast and precise phenotyping is needed. And our institute has invested equipment for application of the high throughput phenotyping tools in practice. For barley breeding, useful methodologies have to still be developed. A trend can be observed when collecting statistical data. Sowing area of barley in Latvia has gradually decreased over the years. Currently, barley is in the 4th place behind winter and spring wheat and also behind oats in last years. On the other hand, spring barley sowing areas are decreasing in proportion to the increasing of winter barley, which reached now 13,000 hectares in last year. Farmers are increasingly choosing to growing winter barley because obtained grain yield is a third higher than for spring barley. They are also ready to take risk because, as you can see, every 4 to 5 years there have been grain yield losses due to unsuitable wintering conditions. And to ensure the stability of harvest, one of the solutions is to offer more winter barley varieties suitable for our local conditions. Some more statistics for your consideration, thinking about the changes we are observing in our region in this recent years. There is a relatively dry period in the first part of May, just after sowing. In Latvia, sowing time is usually in the 3rd decade of April. The ability to form well-developed root system and the beginning of the vegetation period and early vigor are the characteristics that the breeder should pay attention to. We detected the number of seminal roots and root grow angle varied significantly among spring barley genotypes. Yeah. Also in the 1st and the 2nd decade of June, during the period when the productivity of air is forming, a tendency has been observed that the average daily air temperature rises, but precipitation decreases during this period. It could be taken account also for breeders. And summarizing conclusion of barley breeding in Latvia, the focus of the barley breeding in Latvia is centered primarily on the local market, feed and food grain raw material producers, including organic farming sector. In Latvia, barley breeders is also researchers. New barley varieties are developed mostly registered thanks to the realized research-based projects, and cooperation with the geneticists so far has created new knowledge and insights with some useful practical contributions. To speed up the breeding process for the evolution of prospective material, close cooperation between barley breeders is important and should be continued. And previously mentioned changes meteorological parameters observed in recent years opens a greater perspective for broader cultivation of winter barley and proportionally reduce the areas of spring barley. And new selection criteria for increasing the adaptability of the variety should be specified or defined. And thank you all our collaborators. I wish you good luck for everyone, especially for barley breeders, to be able to see the value and potential of their work and to be able, although it's a reality, we are competitors in the market of barley varieties. However, our ultimate goal is common, to make this world better. Thank you.
Yeah, you were mentioning organic breeding for organic cultivation. Do you know how much of the barley grown hectares here in Latvia are under organic cultivation?
Will not know. I cannot say this precise statistical data. In any case, it's rather difficult to find precise. We usually find only through certified seed areas these distribution of varieties and cereals. But you can ask Linda, because she are deeper in this question.
You said that you are developing heterogeneous variety with the help of composite crosses.
Yeah.
Can you tell how do you do the selection from the composite crosses?
Selection. Selection is based on basically it's natural selection. These varieties actually aim of those varieties we are preparing especially for farm, special location, special soil conditions, and during the years we suppose this variety more and more will adapt to local conditions. It's the sense in this case, yeah.
And what happened to the yield over the selection years?
What happened?
To the yield?
To yield. If variety will more and more adapted, it means yield more stabilized,
Yeah.
and it is a benefit for farmer, I think, yeah.
Yeah, okay, it's another
It's
discussion.
our research only in the first stages. Yes, in this now submitted article, we saw that CCPs showed. It also depends from CCPs genetic coverage. Some of them shows higher this yield stability and yield potential. For example, there are also different difficulties. For instance, for Kalles barley CCPs, Ustilago nuda in English. I forgot. It's very big problem for them. It's important for organic farming.
Thank you very much, Søren. Can you hear me? Yes. I would like to thank the organizing committee for giving me the platform of going for an oral presentation here. It was really nice, and also nice to be the presentation that you've all been waiting for, the last one. I would like to say hi to the online audience, because I know I have a friend in Australia, Katrine, who is watching. So now to my speech. So what is unique with Icelandic barley? Yes. So we already heard a little bit yesterday about the climate in Iceland, which is a little bit different from what most of the people here are focusing on. Our climate is quite different. It's very long days. In the summertime, it's bright around the clock, basically. We don't have very much trees, so the wind can gain speed. So we have strong winds. But I think the main problem is the temperature for the barley, because we have only around 75% of the heat sun during the growth season. And you can see this is a graph of the monthly means during the year in Reykjavik, and you can see it barely reaches 12 degrees. So we know this is a little bit suboptimal for barley. But we try as good as we can. So there has been an Icelandic barley breeding program since late 70s, beginning of the 80s. This man is called Jonathan Hermansson, and he was the barley breeder for about 35 years. And he sort of, so we heard a little bit yesterday already about the background of the Icelandic barley, which basically was made from the start, from the beginning of the 1970s, because we lost the old land races in Iceland. There are 2 Faroe Island races that have been used, and they are sometimes assigned like they come from Iceland. But they are from Faroe Islands, and then Jonathan and his predecessors used some exotic material, but a lot of Nordic material, primarily from Norway and Sweden. Also some influx from Scotland. His main breeding goals, besides yield, of course, is the ability to mature in low temperature. This is like quite difficult under these conditions. And along with that, early heading. And then, like I said, straw stability is needed in these strong winds. Not necessarily too short, because the farmers actually also like a bit of straw. But it should be able to withstand strong winds. So what is unique with Icelandic barley? Now I give it away. It is very early. And when I came into this project 10 years ago, we started looking in the sort of population of breeding lines and material and cultivars. And among those, there were 3 genotypes that are extremely early. We also checked this against quite a big number of exotic material that we ordered from gene banks around the world. We have not found anything that is matching its earliness in Icelandic conditions. The closest are to a couple of East African lines that were similarly maturing, but not earlier heading. So it is quite early, at least. So now we wanted to try whether this earliness was also seen in other environments, because it could be interesting for more areas than Iceland, perhaps. So then we had this golden opportunity of this wonderful Nordic collaboration that Therese talked about earlier. The PPP Barley project, Nordic collaboration among all the 5 Nordic countries. And here we had the ability to add our lines into a Nordic gene pool of 180 lines. And we got them screened in 17 field trials. And we also got them genotyped. And then basically we could establish that the earliness is happening also elsewhere. So you see the number of days to heading changes quite a lot, but you always see some very early outliers, and these are the Icelandic lines. They were always at the earliest heading. But then we wanted to find out sort of the genetics behind this earliness. Sort of the main thing we wanted to find was actually markers to predict earliness, and that was maybe a little bit naive considering the complex genetics behind flowering and earliness. But anyway, we tried to do GWAS, but of course 3 early genotypes in a set of after some selections and filtering. It was 169 lines. It was a very low allele frequency. So what Hadourabi, who is here today, and Gunther Backes did long time ago now, was to actually call out the markers that were associated with exactly these 3 lines. So early on we found that there was some polymorphism in the SNPs linked to the PPDH1. Only found in these 3 early lines and 1 of their ancestor lines, which is a Scottish line. But it did not express the early phenotype. And all the other Nordic lines had the other genotype. So then we wanted to try to what happened in controlled environments, because we wanted to see a little bit if we could see any effect of day length or temperature. So for this, we went to Norway and got the opportunity to do greenhouse trials with controlled environments. And here we try to mimic the Icelandic environment in one of the trials, which is called long day cold. So there we set the day length to 20 hours and the temperature to mean 10 degrees, 5 degrees in the night and 15 degrees in the day. And then we took like a really warm climate, which had 20 degrees. So our standards are a little bit different from other people's. So we had a long day warm also, and then we had short days with 12 hours, same cold and warm temperatures. And then we screened these for heading day, maturity day, and also height, which I'm not going to show you. And then we also calculated the heat sums to heading day and maturity day. So, as you can see here, is the overall results of all the lines, and you can see that there is actually no difference, no statistical difference between long day cold and long day warm if we look for the accumulated heat sum. Of course, there is a difference in number of days, but when we look at exactly the temperature, it was the same. So the plants seemed to need a certain number of degree days until they had. In the short day warm climate, you see there's a very wide distribution here, and we had several lines that never headed. And then on the picture here, I thought this would be the best possible audience to actually put this picture here, because this is a loose end. We haven't followed up on this, but I think it's curious. So maybe some of you may have some ideas. So here on the right-hand side, you see the earliest heading line, the 247.11, in long day warm, and then in the middle is same accession in long day cold, and then the left-hand side is the short day cold. It's the same material. So it looks completely different. You have these long spikes and a taller statue, and actually much more vigorous looking. It's very easy to think in retrospective what you should have done, but we never collected the spikes, so we do not know, we do not have data on the fertility of the spikes, which would have been very interesting. But maybe some of you may have some ideas about this. So then we did a GWAS also on the data from this experiment, and we found a few associations, and some of them matched with sort of expected loci. So we picked up a few of those for the next part, which was to resequence 4 genes. So based on those results, we picked out these genes, PPDH1, the ELF3, and the FT1. And then we know from literature that centroradialis gene is very relevant in the Northern European barley germplasm. So next step was to resequence these 30 genotypes that we selected. And we selected these genotypes based on the pedigree, hoping to sort of trace the genetic diversity back in time. And these genes have different names, of course, like all of you knowing, so it has been a little bit confusing sometimes, but this is just the 4 genes. Then we have here, so we didn't have GIL data on all these 30 genotypes, as a lot of them were also historical. But these we had for quite a few of them, and we also plotted then the yield versus the heading day to see the distribution here. And you can see in the lower left corner of the graph, you see the earliest heading line, the 247.11. This is based on quite a few trials. So. And then in the upper right corner, you see another group of Icelandic barley lines and 1 Finnish line. So what was in common for these groups? So here we have the results from the sequencing of these lines, and you see the same graphs here. So for the L3, which has earlier been, it's like the day length neutral lines, and we also identified those in the greenhouse trial. They were very clear, but we didn't see any real clear pattern when it comes to effect on earliness combined with yield in Icelandic conditions. But when we look at the PPDH1, we see that both of these very early lines that were included in the resequencing project had the winter type allele of the PPDH1, sort of the dominant, the non-responsive allele, the responsive allele, sorry. Whereas the normal yielding ones had the recessive allele. And then for the centroradialis, we can see here that the upper left corner had the same allele, and it was unique in these lines. So it was the allele that is typically found in winter barley. But here we see an effect of it in Icelandic conditions. And then for the FT, we did not see any results. So if we plot these in the haplotype combinations of these 4 genes, we see that the earliest heading lines had statistically different or significant difference to the high yielding and early lines. So this was quite curious to see that the centroradialis gene in the spring barley background has this effect that you can actually combine earliness and yield. So just to sum up, this latitudinal distribution of alleles in Europe that has been reported several times is normally stated that the winter barley is grown in the south, of course, and they have this photoperiod responsive allele of the PPDH1. And they also have this type 2 allele of Hovecentra radialis. Whereas the spring barley in the north typically have this non-responsive allele and the type 3. So what we saw here in the north, the extreme north, I should say, was that it seems to go back. So we have actually selected for the responsive allele in the spring barley background in Iceland, and the same with the centroradialis. So the take-home message is that the photoperiod responsive allele creates this extreme earliness in the spring barley background, particularly in this line, 247.11. Whereas the winter type allele of centroradialis has effect on earliness without the yield penalty. So we conclude just that it is quite good to select the material in the target environment. So with this, I just want to say, despite this really quite harsh climate, it is definitely possible to grow barley. And I want to thank my contributors over the years, particularly Thorbjörg Helga, who did this resequencing part of the study. And of course, the whole Nordic PPP Barley project. And these results have all been published in 3 different papers. So you can find it there. Thank you very much.
I'm just curious, have there been black-seeded varieties among the test trials?
Not among the ones that were included in these experiments, no. There have been black-seeded way back in the breeding program, but not in these pedigrees, no.
Perhaps you can combine it like a sun collector on the roof?
Yeah, yes. It would actually be quite interesting.
Yeah, sorry for this naive question, but you mentioned that you have actually not very harsh frosts in Iceland. Why didn't you try any winter barleys?
They have still difficulties. So we can grow winter wheat in limited locations. They are a little bit hardier. But winter barleys seem not to tolerate this unstable, fluctuating conditions. We do always have frost at some point, but very varying snow cover. So I guess that is the limiting factor. Typically the wheat grows reasonably well in the areas where you have a fairly stable snow. So, yeah.
Alan?
Yes, just a comment a little bit off the subject, but if you're talking about what's unique about Icelandic barley, you should mention that the only company in the world making money from transgenic barley is in fact in Iceland, the Orb Genetics.
That is very unique, yes. That's a good point.
And Daniel?
Magnus, when you put your plants under control condition to simulate what happened in the real world, you mentioned that the thermal time units was the same, but when I show you your photograph, the long day cool plants were short with respect to the short days, and suggests to me that there is a photovideo sensitivity between both conditions, and why the thermal time didn't change.
So the thermal time to heading did not change in the long days. We definitely saw a height effect, not in this genotype, but in other genotypes. We saw a height effect in the long days also.
In other words, you have different plant height with the same thermal time units, soon? This is confused for me. Okay. This is not frequent because probably you change the number of leaves that determine different lengths of the internodes or so.
I think I can show you one graph from the paper, and I think we can look at it together. Because there was a lot of variation in responsive in height, yes.
Okay.